Thank you.
Thank you very much, Dennis. It's a pleasure to be at this online journal club, and thank you for the people that are streaming this talk. I'm Gioele Manno from the Stellinerson Lab in Karolinska Institute in Stockholm, and I'm going to talk about molecular diversity of midbrain development in mouse, human, and stem cells. Our paper was published now 6 months ago in Cell. So I'd like to start, since I'm going to talk about embryonic development, I'd like to start by talking about the complexity of the developmental process, in particular looking in a tissue perspective. So tissue is composed by many cells, and they undergo several processes during the embryonic development, going from an undifferentiated tissue to a specified tissue that is anatomically structured and physiologically functional. The series of events that happen in this tissue is complex and is concerted, many things happening at the same time. And that's why molecular biologists and developmental biologists eventually have learned to use some kind of general principles, some kind of pillars to interpret it, dividing this process in a differentiation, a lineage, niche, and patterning. Through this kind of concept, we can address the complexity of this process, and we make eventually experiment in addressing those different parts of the process going on. So we make experiment in differentiation, we study particular aspect, and another one regarding lineage, and then at the end we make a kind of we summarize it, making sense, interpreting it. That's the way developmental biology has been progressing right now. And what we would like to propose with this study that we publish is that one could go about solving this problem in a little bit different way. So that we have our developing tissue, and we could sample from it. That's what we did. Basically getting single cell transcription profile using single cell RNA sequencing, and basically sampling this tissue at different time points until we have enough cells that allow us to make a computational analysis and deconvolve those aspects I was talking about, lineage, differentiation, niche, and patterning, from the same data, from the same cells, seeing this basically happening all at the same time, by deconvolving, looking at those aspects one by one. And we decided to do that in a model in Ventral Midbrain, because eventually we think that this is a really good model system to study neural development. It's an ancient part of the brain. Actually, the relative size of midbrain has kind of decreased over time because other parts of the brain become more and more complex, while midbrain structure remains relatively the same. So interesting in this sense. And there are important clinical implications. The dopaminergic neurons that sit in this part of the brain, when degenerated, are responsible to the symptoms of Parkinson's disease. And therefore also the translational implication of studying this process of differentiation of those neurons, with the hope that one day it would be possible to differentiate, to take cells from a patient and differentiate them first in immune-induced pluripotent stem cells, and then through a differentiation protocol, make the dopaminergic neurons to inject back into the patient brain to try to recover the symptoms and the disease. So this little introduction, and then I'm going to go directly into our sampling strategy. Our experimental workflow is relatively simple. So we start from embryos that we dissect the tissue of interest, the ventral midbrain, and we make whole tissue cell suspension. And we do this for several time points of human and mouse development, ranging from time points between week 6 and week 11 after gestation, after conception, and days 11.5 to 18.5 in mouse. We also have a time point adolescent mouse. And we perform single cell RNA-seq using a fluid MC1 platform that allowed at the time to do 96 cells at a time. And our technology, single cell RNA-seq, allows to make use of the so-called unique molecular identifiers, also called random molecular tags. What those are, they are random sequence, random oligos that are incorporated to the cDNA when their RNA is transcribed, so that we will have labeled every molecule, and then we will read it in the Illumina sequencer. We can actually count the original number of molecules, allowing us to have a measure of the number of original molecules present in the cell instead of just reads. So first thing that we do in such a data set is try to group those cells. So we are sampling unbiased, so we're sampling every cell in the tissue. And we first thing that we need to know is kind of to group them in cell states, or if you allow me to use this term, cell types. So here I'm showing the 26 cell types that we found in the mouse using clustering. I'm showing, so every dot here represents one cell, and this projected in a two-dimensional space, because this data set has a lot of dimension, as many as all the genes that we're measuring. And it's projected into using TSNE at a dimensionality reduction technique that allow us to see cells that are similar close to each other and cells that are different far from each other, and they are colored by the cluster that we named afterwards, looking at gene expression and time distribution. So what you can see here that are cells of the vasculature, pericytes, and endothelial cells of the immune system, microglia, the resident macrophages. We have progenitor cells that are stemming into lineages. On the one side, glial lineage, on the left side, more diverse neuronal lineages, the dopaminergic, for example, GABAergic one. And we have also some neuroblastic population that I'm going to discuss a little bit better later. And we have this kind of data set and analysis for both human and mouse. And we found basically the same number of cell types in the human as well. So first thing that we can do, and that we did, is to analyze the expression landscape to compare a little bit those cell types one to each other. One first thing that we saw is that there was an abrupt change in the transition from progenitors to neurons. So that you can see these boxes like basically progenitors similar to each other, and as well neurons and neuroblasts overall, but there is an important switch happening between the two steps. Another thing that we could visualize was the time distribution of the appearance of those cell types over time. So yeah, I have the time points that we sample at, and since this clustering is done by including all the data sets all together, it's possible to ask later, what is the abundance of every cell type at every time point? And one can see a situation like this, where there is a cell type that is very abundant at the beginning, and numbers decrease over time and eventually disappear. So when other cell types are appearing, that's a clear example of cells that are basically differentiating in more mature cell types. Okay, I've said before that we use a highly quantitative technique that allows us really to count the number of molecules. But RNA expression is noisy, is bursty. And other problem is that when we sample the RNA molecules, we don't have 100% efficiency. So eventually we have dropouts event where there were molecules in the cell, but we don't measure them. So we need to tackle that in a statistical way. And we use a Bayesian negative binomial generalized linear model to study this process. In particular, we model a gene X, the expression of a gene X, as a linear combination of a baseline component that is taking in account for example, a leaky promoter or housekeeping level of expression, and cell type specific components that are taking in consideration only if that cell belong to cell type A, for example. And the sum of those components basically explains the expression of gene X. So I'm showing what we get as an output. We get a posterior probability distribution of the expression levels of genes. And here you're looking at the expression of KDR over the whole cell type in human. And you can see how the distribution of probability, like the expectation and uncertainty around the level of expression of this gene, it's relatively narrow and does not overlap with the distribution of the other genes. So basically we can conclude that KDR is specific for human endothelial cells, and this basal level is basically 0. So we can call almost in a kind of binary way a pattern of expression, where K would be, like you imagine, a vector of all zeros and the 1 in correspondence to endothelial cells. So I'm drawing these gray boxes below it to signify this. And for example, this can also catch significance of many cells, like TH here is expressed in these 3 dopaminergic cell types. And we don't need to do 1 versus all statistics. We just can call the significance because the posterior probability distribution, it does not overlap with the one of the baseline. With this kind of approach, we really built a kind of mini atlas, and here just one figure for the supplementary part of the paper. We can really list a couple of genes in this case, but we have a website on linearsonlab.org where really you can browse and search for your favorite gene and see in which cell type is expressed. It's relatively, relatively complex if you then go into more complex patterns, and but here we really list some of the markers that we think they're extremely trustworthy, and the statistics were really striking. And that's out by the fact that we have absolute count. Again, we count the number of molecules.
Sorry, does this model assume that for each gene there seems to be very few cell types specific? Is there any gene that you will see express uniformly across all cell types, or at least more than 50%, something like that?
Yeah, so I'm not showing that, of course, because kind of a little bit, well, one could argue that it's like kind of a little bit boring, I mean, to show the discovery of the paper. But of course you can, if you browse for, I don't know, actin or tubulin, eventually you find that it will look a little bit, so the pattern will look in that case, a very high baseline. So if the gene is really, there is no dynamics in, the predictors of the cell types don't explain extra component, so the model is automatically kind of regularized. So we'll assign the expression to the baseline, and you will see that that gene is expressed only at baseline. And in some cases, like you said, it's also possible that basically 50% of the cells express it, and like they're called significant. And this happens, for example, for genes that are pan-neuronal. They're expressed in all neuronal cell types, and we have here almost half of the cells are neuronal. So this actually indeed happens. We're just I'm not showing here just here, but
thank you.
Yeah. So progressing on another figure of the paper, we analyzed a group of cells that that we found were basically very similar for overall gene expression, and also hadn't been really kind of studied very well before. The neuroblast in this part of the tissue. They were mostly interested of some neuroblasts, the one that make dopaminergic neurons later on. And what we saw that they were actually really strongly marked by few genes, and importantly, most of them were transcription factors. So we got curious about it, and when we went and looked using in situ hybridization, we found out that what we were looking at was a patterning event that take place in the ventral midbrain, and we characterized, and eventually we were able to draw those areas where those transcription factors were expressed, and corresponding to those cell types. So the ventral midbrain, it's a pattern, but already before the cells acquire specific neuronal signatures, it's already patterned, and the regions are defined by expression of those transcription factors. And we tried to extend also this idea of like studying what is the transcription factor code of every cell type we found. Using this fact that we can make a claim about regulation by the fact basically we can call a statistical significance for one gene and get kind of a binary pattern. We summarized all this pattern, all this call of significant genes in one or more cell type to get a more complex view in some way in a layout that is a little bit familiar in reviews, for example, in developmental biology, where we draw a rectangle here below a cell type and we label it with the name of the cell types that have that pattern, so that one can basically read specific transcription factor specificity. And we don't include only specific transcription factor pattern, but also combinations. For example, A1 that is shared by radioglia 3 and radioglia 1, or SOX4 that is shared by this group of neuroblasts. So we can really read this scheme both vertically, horizontally, depending what we're asking. At this point, I've been kind of described the kind of analysis we do, but I haven't talked about how do our human data set compare with the mouse one. And the kind of analysis that we try to do here is to try to find couple of cell types that are homologous to each other. So this idea of cell type homology. So the way we went about it was relatively conservative and simple. So we selected first the genes that had some dynamics, expressed dynamically either in the mouse or the human. And then we calculated the correlation coefficient between the expression profile of those cells. So
how many, Gioele, how many genes were this, around about?
So this is, we start of course with the 20,000, then we filter and it ends up to be around 3,000 that are significantly expressed, and we use some filters. The problem is here, if you use housekeeping genes and you put them in, what we would see is just kind of the depth of sequencing or very uninteresting axis of variation. So you want to really restrict the number of genes to the ones that have some dynamic. Yeah. What we saw here, what I'm doing here with these lines is I'm drawing a horizontal black line in the maximum of every row and a vertical line in the maximum of every column, so that where we see basically those crosses, what this is marking a mutual best match between the mouse and the human cell type. So basically the mouse is the most similar cells to the human and vice versa. So in this way, we kind of find bona fide homologous cell types. And at this point, to convince you, I will show you some market tables of where on the left, the human dataset, on the right, the mouse. You see the expression of those genes and their homologous. And I'm also as columns, I'm putting the human cell types and the corresponding mouse cell types. So first thing to notice is that the pattern of expression is extremely conserved. And even when actually a gene is not co-significant, but still expressed at a low level, even this pattern, like this lower expression, it's still conserved. So it was quite remarkable, in my opinion. And another important thing that was really unexpected is that even the dynamic range, so if you look now at the y-axis, this is representing the number of molecules that we count. And you can see both the two plots really have the same scale. And you can see, like, for example, for Claudin 5, the level of expression of the Claudin 5 endothelial cell, it's basically the same. It's extremely conserved across the species. And this is, of course, a pick that I made, but we quantified this overall, and we found that there is, with a p-value of 10⁻12, there is an increased correlation of the expression level of the genes that we observe versus a background model. So really, homologous cell type expressed genes at a very conserved level. So at this point, I talk about cell types as a kind of in silico entity. I've clustered the data and I've told you those are cell types or those are cell states. But of course, what we do in the paper is also to validate the existence of those cell types. And one way that we do that for rather glia-like cells that were uncharacterized before as different types, it was using single molecule RNA fish. So the approach goes pretty much like that. We probe single molecules of mRNA by using tiling oligonucleotides that are marked with fluorescent probes. And we can really detect dots corresponding to one molecule of mRNA. Through different cycles of stripping and reprobing, we can then, on the same tissue slice, get the multiple expression of several genes. In this case, we did 3 cycles and we got 8 genes, and we can then fully characterize and basically anchor back on the tissue the discovery of new cell types. That's what we do here. Looking at the ventricular zone of this tissue, you can see already how there is a regional distribution. Like ventrally, only one type of radial glia is present and another type more dorsally. And then you can see some cell types. Actually, here you have radial glia that are differentiating in oligodendrocyte precursors that kind of, we catch them while they migrate into the parenchyma. And eventually they switch the program from this marker, so C6A11 to SOX10. And here also we see these other radial glia cell types that we call radial glia 1. We found eventually that those radial glia types are kind of differentiating to different lineages, astrocytes, ependymal cells, and neurons. Finally, I would like to talk about our effort to determine in a quantitative way the quality of our in vitro dopaminergic neurons preparation. So again, here is in this optic where we hope one day to be able to first take a sample of cells from the patient, make an in vitro induced pluripotent stem cells, and then make do an in vitro differentiation procedure yielding dopaminergic neurons that we can inject back into the patient. But it's difficult to properly evaluate the quality of this preparation, where actually the classical characterization, maybe a little bit the gold standard that you see in papers right now, is to use real-time PCR of a timeline and see the genes that you don't want going down and what genes that you want going up, or using immunofluorescence to try to get a single cell resolution, where usually people is able to detect the co-localization of 2-3 markers at the same time. So we think that this is still not very robust, and it's difficult to compare between labs. We would like to have something really robust and reproducible and quantitative if we want to put cells into a patient's brain eventually. So we went about solving this problem in 2 ways. One is more simple, a little bit of still has some components of the classical characterization in it. That is, we make our IPS differentiation to dopaminergic neurons. We make a de novo clustering of this data set, and we compare pairwise the clustering from the human development, the one that I described until now, versus the one that we obtain in vitro. First thing that we notice, interestingly, is that even if we are targeting, if we are aiming to get 1 cell types, dopaminergic neurons, namely, we indeed get 13 cell types from this preparation. And what also was remarkable is that some of those cell types are extremely similar to things that we see in the tissue, radial glia, progenitor cells, even reduced glutamatergic neurons. Some of them, of course, are target. They're similar to the dopaminergic neuron that we're targeting. But it seems like if you're trying to make a cell type using soluble factors, just signals, basically using ligands, it's what you're still, you can't avoid pretty much to produce cells that are similar to the one that you find in normal tissue, because you're basically copying morphogen induction that happens in the real tissue. So this was quite interesting, we found. And of course, then you can move on and take the cell, say, okay, those cell type IDA, A, B, and C make up for 30% of the culture. They seem quite good, and then you can look now in an unbiased way by selecting manually the genes. You can check and compare with how the cell types look regarding their expression profile of those genes. You can compare to the human reference data set and see, okay, this DAC seems the one that matches more, but there are some differences. This retains still 1 gene that earlier, or is missing some maturation gene. So you can really get an answer what is missing and what is good. But it's not still really single cell, right? Because we are clustering the cells and we are comparing the cluster. We're comparing the average, sort of. And what we really want to do is to give a quality measure on every single cell. So we devised this alternative approach that we call prototype analysis, where we first, using a machine learning algorithm, particularly a very simple one, a regularized logistic regression, we learn a function that is able, when inputted, when we give us an input, single cell expression profiles for the in vitro differentiation, any kind of single cell RNA-seq profile, is able to output a probability that cell is one of the cells, any of the cells that I've been learning about, that I found in the reference data set. So basically, it's giving me for every cell type that I know, it's giving me a probability of that cell to actually be that cell type. And that's a little bit how it looks like. So if I plotted in this wheel plot, that's the way we like to plot and visualize this. Every vertex here representing a prototype, a cell type class that we learned using our machine learning algorithm. And our function, what it's doing is placing new cells, so cells that we are giving to the model, is placing them on this wheel, putting it extremely close to the vertex in case of the probability of them belonging to that class is very high. And that's what it looks like if we put in, we score the cells, the input data set, the in vivo cells, and that's, for example, how it looks if we give a randomized control, basically cells that don't exist, that we obtain by scrambling the data expression profile from different cells. So here the model says, okay, I never saw these cells, the probability is 0 to be any of those prototypes, and here is clearly able, as you see from the colored, the correspondence colors vertex, is able to score them correctly close to their prototype. So now what happens if we apply this? This is just an example, but what happens if we apply this in the real data set? Now I'm showing you this differentiation timeline going from the day 0, the stem cells, up to day 63 of differentiation, and showing them colored from red to blue. And you can see how basically cells kind of move over this wheel, starting from a stem cell vertex to a more general progenitor vertex, and then going to a prototype of the floor plate, neuroblast, and eventually towards dopaminergic neurons. And while many cells are here in this area that is relatively close to different kind of vertex of neurons, in particular dopaminergic ones, only relatively small amount, it's really, really at the vertex. And some of them even go towards out of target effect. So, okay, this is a nice representation, but if we want to even know more about it, we can get an evaluation at the single cell level, but this time using a selection of genes that is not biased, is not decided by what we know from the literature, but is unbiased. So the function that we learn, the machine learning model, is able to actually, if we select the genes that have higher weight, to give a list of genes that best scores, best discriminate the cell type of interest from the others. In particular, if we have here the genes for the scores best dopaminergic neurons and genes that scores best progenitor cells, and in the column we have the top scoring dopaminergic cells in the embryo and in the stem cell in vitro, and the same thing for progenitors in the embryo and the stem cell in vitro. So you can see really at the single cell level, you know, consider this is relatively noisy data, that's how single cell data looks like. You can see how similar the cells are, but you can still see that some modules are kind of missing from the stem cell preparation. Well, for progenitors, we achieve better goals. So maybe the take-home message from this experiment would be that we achieve a good progenitors, but we need to improve in the maturation step of the differentiation protocol. We eventually opted this kind of evaluation, quantitative evaluation scoring technique for differentiation protocol would be used as a gold standard in the field to give a better quality and also make the technique really reproducible. And maybe, you know, we could envision a situation where we really send our cells to a co-facility and we get the scoring that is unbiased, and so it's in this control and robust over, you know, different labs in different countries, for example. So at this point, I think it's time to conclude. I hope that I've been able to show you how in our study we achieve an unbiased classification of the cell types in the human and the mouse ventral midbrain. And we define them molecularly, providing a kind of mini atlas. We found new cell types, in particular radioglial cells, and we offer also a species comparison of cell types in the two species, define cell type homology, and showing how the dynamic range of the expression level is extremely well conserved across these two species. And finally, I show you how our machine learning scoring approach to use what we learned from the in vitro, from the in vivo data, and applying the in vitro differentiation protocol to get a better scoring that is quantitative. So with that, I'd like to thank the people that work with me, in particular Daniel Gilborg, that is the first author of the paper together with me, and all the Linnerson lab, Simone, Amit, Lars, and also Arena's lab and Barker lab that has helped us for the sampling of the human. And to you for listening. Thank you very much.